STB1093 Microbiology Experiment 3 Determining numbers of microorganisms in a sample, presented by Group 6, Subgroup 1.
Determining the quantity of microorganisms in a specific sample is frequently crucial. Several techniques have been devised by enumerating bacterial populations in laboratories with a standard plate count, turbometric method, and direct microscopic count being the most commonly employed methods. Each of these approaches has its own set of advantages and drawbacks. There are 4 general approaches used for estimating the size of microbial populations. Cell numbers can be determined through direct counts. Besides that, we can also determine the cell mass, as the biomass can then be related to cell numbers through the reference of a standard curve. Cell numbers can also be indirectly determined by diluting or concentrating microorganisms in a sample and cultivate them on an appropriate medium. Another indirect method we can try to determine the cell mass is by measuring consistent biochemical elements within microbial cells. The prevalent technique for counting bacteria is the viable plate count. This approach involves creating serial dilutions of a sample containing viable microorganisms, which are then placed on a suitable growth medium. The suspension is either spread over the surfaces of agar plates or blended with liquid agar, poured into petri plates and allowed to solidify. The plates are then incubated under conditions conducive to microbial reproduction. Observation of colonies can be done without the need of a microscope. The assumption is made that each bacteria colony originates from an individual cell that has undergone cell division. Hence, by tallying the colonies and factoring in the initial dilution, it is possible to compute the quantity of bacteria in the original sample.
3 of 99 ml dilution splunk were labeled as 10-2, 10-4, and 10-6 respectively, while the 6 petri plates were labeled from 10-1 through 10-4. The unknown sample was shaken to ensure an even distribution of microorganisms, and 1 ml of the sample was aseptically removed with 1 sterile pipette and transferred to the 10-2 dilution splunk. Next, using a new sterile pipette, 1 ml from the 10-2 dilutions was transferred to the agar plate labeled 10-2. Next, another volume of 0.1 ml from the same dilutions was transferred to a different agar plate labeled 10-3. Additionally, 1 ml from the 10-2 dilution was transferred to the 10-4 dilution splunk. From these 10-4 dilutions, 2 different volumes were aseptically transferred to a specific agar plate. Using a new sterile pipette, 0.1 ml from the 10-4 dilutions was placed onto the agar plate labeled 10-5. Another volume of 1 ml from the same 10-4 dilutions was transferred to a different agar plate labeled 10-4. Melted agar at 50 degrees Celsius was aseptically poured into each petri plate to which a sample dilution had already been added. The plates were swelled to mix the sample with the agar without letting it run over the edge. The lids were replaced and the agar was allowed to cool and solidify. Inverted plates were incubated at 30 degrees Celsius. 9 ml dilutions water blanks were labeled as 10-1, 10-2, and 10-3, while 4 nutrient agar plates were labeled 10-1 through 10-4. A 1 ml portion of the water sample was aseptically pipetted into the 10-1 dilution tube. The 10-1 dilution tube was truly mixed by vortexing or vigorous shaking, resulting in a bacterial concentration 1/10 of the original sample. Using a new pipette, 1 ml from the 10-1 dilution tube was aseptically transferred to the 10-2 dilution tube, followed by vortexing. This created a bacterial concentration 1/100 of the original sample. 1 ml from the 10-2 dilution tube was transferred to 10-3 dilution tube and mixed, resulting in a bacterial concentration 1/1000 of the original sample. Next, with a new sterile pipette, 0.1 ml from each dilution tube and the original sample was aseptically transferred to agar plates. This additional transfer effectively dilute the sample by another factor of 10, ranging from 1/10 to 1/10,000 of the original concentrations. Starting from the greatest dilutions, 0.1 ml was pipetted onto its respective agar plate. The glass spreader was sterilized by dipping it in alcohol and then flaming it in a Bunsen burner flame. Once cooled, the sample was spread over the surface of each plate by touching the rod to the agar and rotating the plate. This spreading procedure was repeated for each plate, ensuring the sterilization and cooling of the glass rod before spreading each sample. The inverted plates were incubated at 30 degrees Celsius for 24 to 48 hours. After incubations, colonies were counted on plates within the 30 to 300 range and counts were recorded. The concentration of bacteria in the original sample was calculated based on the count obtained from the dilution plates, considering the dilution factor applied during the process.
This is the result for experiment 3A, pour plate method. None of the experiment results can be used in the calculation, as all of the data is below 30. This is the result for experiment 3B, spread plate method. Only the result for 10 to the power of -1 dilution can be used in calculation, as the data for the others are above 300. Discussion. It is difficult to directly count the number of microorganisms in a population. Instead, we estimate the number by counting a sample. The variable plate count is a common method used to enumerate bacteria. It involves spreading a sample on a neutron agar plate and counting the number of colonies that grow. Each colony arose from a single viable cell in the original sample. While useful, this method only counts viable cells that can form colonies. It also takes time for colonies to develop, and overcrowded plates can lead to counting errors. To solve overcrowding, the sample can be diluted before plating. The spread plate method allows for the enumeration of both heat-sensitive and aerobic microorganisms by inoculating the sample on the agar surface. In contrast, the pour plate method involves pouring the molten agar into the sample in a petri dish. This exposes heat-sensitive microorganisms to potentially damaging temperatures. For experiment 3A, pour plate method, our experiment results are 28, 27, 12, and 5 visible colonies in the 10 to the power of -2, -3, -4, and -5 dilutions respectively, which are too few to accurately calculate bacterial concentration as they are all below 30. Statistically, only counts between 30 to 300 colonies are suitable. This might be because the issue with the pour plate method is that the hot agar can damage or kill heat-sensitive bacteria when poured over the sample. To prevent this, either the agar can be cooled but still molten before pouring, or the spread plate method can be used instead. Overall, our results show that no visible colonies form when using the pour plate method. A potential reason for this is that the agar traps the aerobic microorganisms, preventing oxygen from penetrating into the agar. Since aerobes require oxygen to survive, they likely fail to grow without sufficient oxygen diffusion into the agar. The lack of oxygen diffusion and the entrapment of aerobes in the center of the agar cause the aerobic microorganisms to die, resulting in no colony formation on the plates. Experiment 3B, spread plate method. Our spread plate method results show 155 colony forming units CFU in the 10 to the power of -1 dilution, and too numerous to count TNTC colonies in the 10 to the power of -2, -3, and -4 dilutions. This gives an estimated mean of 1,550 viable cells per milliliter. In the spread plate technique, the bacterial suspension is poured over the agar surface and spread evenly using a sterile glass rod. This allows the bacteria to grow on the agar surface rather than being trapped inside like the pour plate method. The spread plate technique avoids damaging or killing heat-sensitive bacteria. Comparing the pour plate and spread plate results shows the spread plate method yielded higher colony forming unit counts. This is because the pour plate method can damage or kill heat-sensitive bacteria, resulting in lower cost. The spread plate method does not have this issue. Therefore, the spread plate CFU counts were higher than the pour plate counts due to the better viability of heat sensitivity bacteria. Question for Part 3A. The first question is, why are counts above 300 and below 30 statistically unreliable? This is because counts below 30 are more subject to random error. With such a low count of colonies, natural variation can have a big impact on the count. On the other hand, counts above 300 increase the risk of colony overlap and merging, making accurate counts more difficult. Dense groups of overlapping colonies become hard to precisely enumerate. The second question is, did you enumerate all the bacteria using this method? The answer is no. The third question is, what factors make the pour plate method selective? How can you reduce the selectivity of this procedure? The first factor is the temperature, where incubating plates at different temperatures can select for mesophiles, thermophiles, or psychrophiles. This selectivity can be reduced by incubating plates at moderate temperature of 30 to 37 degrees Celsius. Also, avoid extreme incubations that select for thermophiles or psychrophiles. Another factor is atmosphere, where aerobic and anaerobic conditions affect growth of oxygen-loving or oxygen-hating bacteria. This can be reduced by using aerobic incubation for non-fastidious organisms. Anaerobic incubation introduces selectivity. The last question for this part is, how can you achieve a 1 to 100 dilution other than by adding 1 ml of sample to a 99 ml dilution blank? This can be achieved by taking 1 ml of a 1 to 10 dilution and add it to 9 ml of diluent. This makes a two-step 1 to 10 ratio followed by 1 to 10 ratio dilution again to achieve an overall of 1 to 100 dilution. Also, another choice is by adding 10 micrometer of sample to 990 micrometer of diluent. This maintains the 1 to 100 ratio but uses smaller volumes. Besides, for solid samples, add 1 gram sample to 99 ml diluent. The 1 to 100 ratio mass to volume ratio also achieved. Now we will move on to question for part 3b. The first question is, what does the term colony forming unit mean and how does it differ from a colony? A colony forming unit CFU refers to an individual viable bacterium or fungal spore that is able to divide and form a single visible colony on a plate. The difference between a CFU and a colony is that a CFU is a single viable cell that can reproduce to form a colony. It is the founding parent cell, whereas a colony is a visible cluster of microorganisms that all originated from a single CFU reproducing on a plate. It represents the progeny of a CFU. The second question is, what types of errors would be introduced by using a single pipette for all operations in this procedure? The first error is carryover contamination, where bacteria or residues carried over between samples or dilutions could lead to cross-contamination. The second error that might occur is dilution error, where incorrect dilutions prepared due to contamination or pipetting inaccurate. Another error that could occur is calibration drift, where the pipette could get out of calibration with repeated use. The third question is, what are the advantages and limitations of the spread plate method compared to other enumeration procedures? The advantages are allow colony isolation and pure culture techniques where individual colonies can be big, generally high colony counts per plate compared to pour plate method, and less time and equipment needed compared to membrane filtration method. The limitation is not as quantitative as membrane filtration method where the exact sample volume is filtered. Besides, it is more reliant on pipetting accuracy. Also, it has higher detection limit compared to techniques like MPN tubes. Besides, spreading technique can introduce variability. The last question is, how could you modify the spread plate procedure to enumerate obligate and errors? This can be done by before to inoculation. Plates could be placed in an anaerobic jar with a gas generator pack to remove oxygen and create an anaerobic environment. Another way is by using a sterile syringe to dispense dilutions into petri dishes before quickly covering to exclude air. Also, use a reducing agent like cysteine or sodium thioglycolate in the agar medium is also another way.